together. Right. Just make sure that the sound is off so we don't get an echo. And then Teresa, if you want to share the link again so everyone has it.
Oh, the link?
Yes, because people who come in later can't
see
what you posted to me earlier. Brilliant.
Sure.
Welcome to Neuroccino on this Monday morning, 9:31. Today we have Teresa, who's a visiting student from Trento, working with Fabrice here in Bordeaux. And she's going to present for the first time for us at Neuroccino a paper that she thought was exciting and is happy to discuss it. Before we dive into it, she shared the link. I'm going to put it on YouTube in a minute. But just to remind you of the rules of the game, anyone is welcome to join us on Monday mornings for the Journal Club and present a paper, a book chapter, a book, or even a YouTube video they thought was exciting and worth discussing. The only rule is it's not your own work that you come and present. And that's it, basically. So, Teresa, if you want to take it away and share your paper in full screen, please.
Yes. Okay. So actually, I prepared a brief presentation because I thought it was easier for you to follow. Otherwise, it's a bit maybe complicated. I hope it's okay.
That's okay. If you just want to play the presentation, yes, that'd be great.
You see it?
Thank you.
Okay, perfect. So welcome everybody, and today I'm gonna take you in this journey exploring the brain-gut axis, and I hope you will find it interesting. So, okay. So the title of the paper I am presenting is Differential Co-Expression Networks of the Gut Microbiota are associated with depression and anxiety treatment resistance among psychiatric inpatients. Okay, so first of all, I want to brief outline what the gut-brain axis is, or what the brain-gut axis is. So it is a system which comprehends a two-way communication. So we have a top-down communication connecting the brain to the gut, and a bottom-up communication connecting the gut to the brain. In this system, we have different characterizations, different pathways that are involved. We have the action of the HAP axis. We have different endocrine pathways. And moreover, we have bacteria, which are present in the gut, which produce metabolites that are able to act directly on the brain. And at the same time, these bacteria are able also to act on a local level, connecting to the local nervous system, such as the vagus nerve. So, for example, in the figure on the right, I hope you can understand a little bit more what the characteristics of this brain-gut axis are. So, for example, we can see like different bacteria present in the gut that are able to produce some precursors that instead are able to travel through the bloodstream, going to the brain, cross the blood-brain barrier, and, for example, creating neurotransmitters. Or we have a direct action through, for example, glutamate with the vagus nerve. So we have like a very complex situation here. Okay, why is not going? Okay. So the main, let's say, purpose of this system is to maintain homeostasis. And moreover, this system is able also to modulate the immune system. When we have an imbalance in the community of bacteria in the gut, so it can be an imbalance of numbers or related to the species, we can talk about dysbiosis. Dysbiosis can be caused by different things. For example, can be due to genetic, to diet, or to the exposure to stress. But what we know is that dysbiosis can cause a lot of different conditions, such as, for example, a state of inflammation. And it is also associated with the severity of some psychiatric disorders, such as depression and anxiety. So coming back to our paper, the aim of the paper was to identify the gut microbiome biomarkers for treatment resistance among psychiatric inpatients, and in particular in patients suffering from depression and anxiety. So how is possible to identify the microbiome biomarkers relevant to psychiatric conditions? The authors in the paper tried this analysis, which is the Differential Co-Expression Analysis, the DiffCoEx, which is able to find module of genes whose co-expression patterns can vary across the conditions, but we will see in a minute. Okay. Oh, what is that? Okay. So as a sample, they had 100 patients, and just one note: inpatients mean that they were patients that received treatment during they stay in a hospital. So they stayed in a hospital receiving the treatment. And of these 100 patients, 54% were resistant to depression, and this was measured through the PHQ-9 questionnaire, and 43% of the individuals were resistant to anxiety, and this was measured through the GAT-7 score. And then there was a very little percentage of patients who had like specifically resistant depression or resistant anxiety. In this case, they were like considering the comorbidities or resistant depression and anxiety at the same time. So most of these individuals had extensive histories of outpatient and inpatient psychiatric treatment before this study. And so there was a self-collection fecal swab from these participants shortly after their admission. So around like 20 days after the hospital admission. And the authors used this DiffCoEx analysis, which is able to identify modules of microbial features, and they used the hierarchical clustering and permutation testing in the algorithm for calculating the dispersion values across each module and between each pair of modules. Okay. So what about the results? So the general result was that they found a high amount of overlap between the microbial biomarkers for depression treatment resistance and anxiety treatment resistance. And then they further went to investigate that using the common outputs for network analysis. So now I'm gonna go a little bit into the details. So maybe it's a bit tricky to follow this part, but I will try to be as much as clear as possible. So regarding the depression treatment, they used this DiffCoEx analysis, which was able to identify 3 different modules. So what are these modules? These modules are related to the clusterization of bacteria species with a strong correlation between each other and between the patient groups, with a differential correlation between the different treatment resistant patient groups. So they used a binary variable, so depression treatment resistant, which was equal to 1, or depression treatment normal outcome, so responders groups, which was 0. Then they used these identified 3 modules to see the relationships. And among 10,403 bacterial features that were analyzed, they found that 44 features were strongly correlated in the depression treatment resistant patient groups. And maybe it's not so clear to see, but in the first like square on the right, you can see this like strong correlation with the treatment resistant group. And in the other 2 modules, so the like blue and brown modules, they found the opposite pattern. So like a correlation with the responders group, let's say. Okay, so after this, they did the same thing with the anxiety treatment group. So also in this case, they analyzed 10,403 bacterial features, and the DiffCoEx analysis were able to found 2 modules in this case, one including 114 genetic features, and the other one 43 features. In this case, they found that the first module, so 114 features, were strongly correlated with the anxiety treatment resistant patient group, while the other module had the opposite trend. Then, due to the high comorbidity between depression and anxiety, as we saw before, they decided to determine the common microbial features that were associated to these 2 disorders. And so they compared the features that they found to be strongly correlated in the depression resistant group and anxiety resistant group. And as a final result, they found 43 common genetic features. After this, they use PubDator, which is a tool that allowed them to find in the literature the relationship between these genetic features to bacterial and functioning of these bacteria. And they found that in the 42% of the features that they identified, they had more than 15 annotations. So this means that the bacteria that they found had been previously investigated in the literature, and the remaining 58% of the microbial features that they found had fewer than 15 annotations. So this means that they can be newly identified biomarker that need further investigation. Okay, so after this part, they used Cytoscape, which is a software which is able to visually analyze the networks among the bacterial species that they found. And they didn't go further into the details of this analysis, but I think that they just wanted to do this part of analysis in order to show us that these different bacterial species have indeed interaction between each other, and maybe it would be interesting to analyze more which kind of interactions, which are the relationship between these communities, etc. Okay, so coming back to the final part, so the present study identified the co-expression patterns of the gut bacteria associated to depression and anxiety treatment resistance. So they found 33 bacterial features that were associated both to the treatment-resistant depression and the treatment-resistant anxiety. Then also I wanted to show you the notable taxa that they found. I'm not gonna pronounce the name of the bacteria, but they found these taxa that have been previously associated into the literature to dysbiosis, to inflammation. And so, of course, it would be interesting to investigate further the role of this bacteria taxa, specifically related to psychiatric conditions. Future questions can be related to the mechanisms that are responsible for the disruption in the brain related to depression and anxiety, because of course we don't know the direction, the causal direction, or the specific role of this dysbiosis in psychiatric conditions. And another important future question can be, how can we intervene? How can we treat this, or other way? Can we modify anxiety or depression states, intervening in the gut? Okay. So just to end this presentation, I want to highlight some limitations of this paper. So first of all, the psychiatric inpatients that they were considered had different comorbidities, including personality disorders, and personality disorders can be associated with different behaviors, such as the substance abuse or alcohol intakes, or different kind of specific diets. So all of these things can influence the structure and function of the gut microbiota, and this was not taken into consideration. Secondly, when the patients were admitted into the hospital, before the fecal swab collection, they had 20 days in which they were kind of free to follow their specific diet. So of course they were in a hospital, so they couldn't eat what they wanted, but they had kind of freedom in choosing some food, and these individual food choices were not tracked. So this can be important. And then, of course, they had just one single fecal sample. But apart from these limitations, I think that this paper is very important because it gives us the idea that it's possible that some psychiatric conditions can be related to the gut, to the microbiome. And so it highlights the importance of different parts of our body in order to have a healthy brain state. And that's all from my side.
Lovely. Thank you very much, Teresa. Very interesting run through. Any questions in the room or over on YouTube? I'm happily picking them up and bring them over here. Not yet. Let me check over here. In the meantime, I can ask you some questions if you don't mind.
Sure.
So the first question for you is, why were you particularly interested in this study?
Oh, well, yeah, it's not my main topic during my PhD. So, well, actually, I'm quite interested in anxiety disorders. So everything which is related to anxiety disorders, so possible relationship, possible causes, it's very interesting to me. So I wanted to deepen this topic, because yeah, normally when you talk about anxiety in particular, also depression, but anxiety in particular, you don't consider these aspects. So you just consider the brain. So anxiety, something is wrong with the brain, and we have to intervene directly within the brain. So I think that is quite curious to change a little bit this perspective. So considering that maybe we can also have interventions with different parts, and with the gut, I think it's quite astonishing if you think about that. Yeah, and that's the reason. I mean, I wanted to deepen this topic because I think it's very interesting.
Great. The second question I had for you is, so if I got this right, they used 100 psychiatric patients.
Yes.
Do you have a power analysis in the sample or an effect size estimate? Because that seems like quite a small sample size given the multitude of measurements they looked at.
Yeah, I don't think so. Let me check the paper.
And especially for depression, I would assume that we have larger sample sizes available.
Where is that?
Don't worry if you can't find it.
I cannot find right now.
It's okay. Have a look later. Meanwhile, Michelle has a question for you as well.
Sure.
Thank you for the presentation. I was wondering, is there like some brain areas that are more sensitive to the effect of these, bacteria? Is there something in the literature showing that, or?
You mean specific brain areas that are implied in this gut brain?
Yeah, so will change
Honestly,
activity or the sickness. I don't know.
Honestly, I don't know.
Okay.
I don't know because I'm like quite new to this topic, so I didn't have like the occasion to go deeply into the literature. But I think it is a bit like tricky to directly check for specific brain areas that are implied with the gut, because as I like showed you in the first slide, we have like very different functions, very different actions that the bacteria in the gut can exert in the brain. So we have the production of metabolites, precursors, or metabolites, cytokines. So cytokines can interact with the, for example, HSP axis. So I mean, it's so complicated and so vast that I think it might be difficult to identify specific areas. But one thing is that, I was reading a paper that was related to the HP axis, for example. So also here, it was not so clear the relationship between the gut and the HP axis. But indeed, the authors in this paper were saying that there is kind of effect of the gut in the hypothalamus, in the overall production of cortisol. So maybe that's a partial like answer to your question. With the hypothalamus, there is a relationship, but more than that, I cannot tell.
And then most importantly, what should we eat or not eat to feel better about ourselves?
That's the biggest question. I would say a lot of probiotics and prebiotics, so fibers, vegetables, but
no steak.
The good old classics.
All right, thank you very much.
But it kind of brings back the saying, you are what you eat. So if you
exactly.
You have a healthy gut microbiome, you should have a healthy brain.
Yeah, but also in this case, it's difficult to tell, because sometimes you have like people which are perfectly healthy and they eat whatever they like, and on the other side you have people that are very careful following specific diets, but still they have a lot of problems. So
but I guess the question is also about timing and mechanisms. So is this a matter of you can actually influence the symptoms of your depression or your anxiety by changing your diet, or is having the wrong diet making you more liable to then develop psychiatric symptoms and diseases? And the other question is, what is the mechanism? Is it the brain sending down signals messing up your microbiome, or is it actually going the other way around, where your microbiome influences your neurochemistry in your brain?
And probably both.
Now the causality is a big question.
Yeah,
so
because a lot of the analysis you showed was based on correlation, right? So we don't actually know.
Yes. Yeah.
Maybe you eat what you are.
Sorry?
Sorry, I didn't hear that.
Yeah, because you said you are what you eat, but maybe you eat what you are, and that also.
Oh, yeah. True. Yeah, we don't know. We don't know for now the exact direction. Can be both ways, most probable, but we still need investigation in this topic.
But it is an interesting topic, and I'm by no means an expert on psychiatric disorders or the gut microbiome. But I remember reading somewhere that there is a hypothesis that even diseases like Parkinson's, Alzheimer's, they might actually start as inflammations in the gut and then propagate to the brain many years later. Was that also discussed in the article?
Was that? Sorry?
I apologize if I quoted it wrongly, but this is what I remember from reading it.
No, I mean, it's what you said is true. Yeah. There are many studies, maybe more than the studies related to anxiety, depression. Related to some conditions like Parkinson, Alzheimer's, also like autism spectrum disorders. It's a very huge topic, and I think in the future we will go into details in this, because apparently it's very, very important. And for example, for Alzheimer patients, right now, one of the things that doctors suggest is to carefully have control the diet, because that's super impacting on the brain. But
I mean, just dig out all those common sayings, but an apple a day keeps the doctor away, so I guess this is also true.
You're right. Yeah.
Right. Super interesting. It's going to be, I think, throughout our careers and lifetimes, I think this will be quite an exciting topic of research that will yield novel results that will definitely change the way we eat or appreciate our food, I guess. If there is no other questions, we're actually perfectly on time today. And I thank you for presenting, Teresa, and thanks for your question and being here early this morning. We have another Neuroccino next week, Monday morning, 9:30. And also next week, we have a CNS talk. So do join us for that double bill next week. And until then, have an exciting week in science. Bye.
Thank you.